task. People had set up the laptops and your configurations, so Linux installation वगैरह सब कर लिया था. You got through the process successfully? Okay. That's good. But any previous experience of coding, programming, any kind of languages, Stata, SPSS, थोड़ा बहुत कहीं यूज किया हुआ है? R यूज किया हुआ है ना? बस R Python यूज किया हुआ है. That will come in handy definitely, ना? And one thing about the bioinformatics community is that they are a very closely knit community, and they're very open in sharing all their work. In terms of as opposed to जो especially doctors जो होते हैं ना, वो अपना हर चीज छुपा के रखते हैं, किसी के साथ शेयर नहीं करते कि चीज बाहर आ जाएगी, निकल जाएगी. They're like उन्होंने कोड लिखा और GitHub पे डाल दिया. Anyone can go and run that program and replicate the same results. And this is what actually the science is about. Science is about replicability. जो काम आपने किया है वो दूसरा भी उसी तरह reproduce कर सके. अगर वो नहीं कर सकता तो then there means कि there is a flaw in your experiment. सही? Either या तो conditions different थी जिन conditions में वो experiments किया गया. Yeah, if not, your findings are not replicable. So there was some flaw in the experiment. No? So there's one thing in epidemiology which we know as Bradford Hill's criteria for causality. सुना है किसी ने इसके बारे में? Causal criteria होते हैं ना? So causal criteria के अंदर होते हैं कि you have to fulfill a lot of criteria to finally conclude that one things lead to another. Okay, and that implies कि उसके अंदर आपने replicability भी हो, reproducibility भी हो, multiple studies भी जो है वो same direction में evidence दे रही हो, and there should be some biological plausibility as well. And all of these factors then contribute to this causality argument. No? Zoom pe?
Please not one host.
We have people on Zoom. It's just me and Zoom. And secretary.
Well, the screen is there. Nobody's here.
Yeah.
I usually prefer to stand.
Okay. All right. It's my pleasure to introduce Dr. Zahra Hasan as the plenary speaker. And Dr. Zahra Hasan is Ghulam Ali Hirji Endowed Professor in the Department of Pathology and Laboratory Medicine at Khan University. She's a consultant at the section of Molecular Pathology and Laboratory Medicine, and also director of the Faculty of Health Sciences PhD program. So if you're interested in applying for a PhD here, so you can get some free advice here as well. And she herself has a PhD from Imperial College of Science, Technology and Medicine in UK, and her research focuses on both host and pathogen aspects of infectious diseases, with a particular interest in tuberculosis and COVID-19 genomics and mechanisms of drug resistance. At the clinical laboratories, she works to enhance capacity for molecular diagnostics, including those related to infectious diseases, molecular oncology, immunology, clinical genetics, and genomics. And today, she'll be talking about the power of pathogen genomics from molecular epidemiology to signature-based predictions. So thank you, Dr. Zahra. Over to you.
Thank you, Dr. Imran, and thank you for inviting me to the workshop. Thank you for coming. Thank you to all the organizers. It's a pleasure to be here. And I think that you have, 2 or 3 days of a workshop? 5 days. So you have seen machines, you've been in the lab, you've done some hands-on work, a lot of nitty-gritty about how to handle DNA. So I won't be doing any of that. I leave that to the experts. I have been interested in looking at bacteria and host responses from the perspective of genetic changes in both. Today, I will focus essentially on pathogens and touch upon a couple of things that will loop back to how you can explore your organisms of interest, and we can think about what ways this kind of data can be useful to you. All of you are people who are here because you probably have your favorite bugs, and you know what you want to do. Perhaps you want to look at variations after treatment or after vaccination, which is one of the things that Dr. Imran and his team do. And in fact, you're here finding Nemo because it's actually part of a project, as I understand, that's looking at strains of pneumococcus in the context of pneumococcal vaccinations that have been rolled out. But there are a few things I'd like to cover. One of them is to have like a bigger picture as to why do we want to study molecular epidemiology and what is the value. I'll be using SARS-CoV-2 for various examples of how it can help us guide or change our response in terms of management of disease or changes in the way we vaccinate or treat, using what we did in our lab and at AKU as an example. And finally, we'll talk a little bit about how we know this is very expensive business. So how is it that you can use information that is gained from larger scale genomic studies to make something that is applicable, tangible, and useful in your own systems? So overall, why are we doing this? We know that we are part of a continuum where diseases have been around through history. Even when we couldn't describe them, they existed. They impacted populations and subsequent generations and a lot of different civilizations. And then medical science came along in different stages. First, the identification of different organisms, then new ways to manage the infections through new treatments, and then, of course, vaccinations. But the questions, related to why it is important to understand the impact that pathogens have had go back to, the Stone Ages, literally, because we have information about the, early man, the Neanderthals that were quite evolved in the sense that they were able to hunt, they were able to gather, to make paintings, and yet they got wiped out. And developments in archaeology and investigations where now people are going back and finding remains such as skulls and examining them. You're sort of working back to see how big their brain was and how competent they could be. But most likely, they're an example of a population such as, close to us, we have the Indus Valley population that got wiped out. Now, why did these populations get wiped out? It could be an environmental calamity, but it also could be disease, right? And then, the Egyptian civilization as well. Okay, very, very important in so many ways. They built what they did. They developed structures. They developed social societies, they developed trade, they developed so many medicinal things and, warfare. But among the many things that affected them, one of them was plague, okay? And this is a recurrent theme. And then the idea that diseases traveled. So a lot of the way that civilizations were discovered and also impacted was through travel and trade, because you had, and then the maps, the earlier maps, so this is a map from the 1400s, 1482, which is different from the way we see the world now, because it was made by people living in the Northern Hemisphere. So the focus is on the countries in the Northern Hemisphere, which is mainly Europe, and Africa is seen as smaller. So this is because this is how people saw the world. But there was transference of populations from the Romans, who did trade with Africa and trade with the Far East. And then there were documented pandemics from the 2nd century onwards, because it was infections that were spreading beyond borders, beyond infections prevalent in certain areas, okay? And one, of the old world diseases, the bubonic plague, which is caused by Yersinia pestis, which is carried by fleas that infect rats. This was continuing to be a source of plagues in the populations, and multiple times. So it's documented in the 2nd century, and then more later on in the 1400s in Europe. So large populations were killed because of, for example, the bubonic plague. And then, global changes in civilizations occurred, because when one country or the invaders discovered other continents, so for example, the discovery of South America by the Spaniards was also very important. The Spanish conquest, which started in the 1500s, they discovered South America, and they brought with them new diseases. And they were able to wipe out the Inca population, which was a very well-established, very well-developed population. They had, civilizations that had existed for centuries, but they were unfamiliar. Their immune systems could not combat the new infections that were being brought. So in a sense, our need to understand infections and how they're transmitted, how you can prevent them, and has been relevant through the ages. And I guess you would call it old biological warfare, without that, maybe on purpose or accidental, but that's how it worked out. So you have classically, this list I thought would be interesting to share, the top 10 old world pathogens, which start with bubonic plague, smallpox, yellow fever, TB, measles, malaria, typhoid, influenza, typhus, and chickenpox, in that order, in terms of infections that have caused most devastating impact across populations in the world. And for those of you that have been studying infections and infectious diseases, it's important to consider this, because this is how invaders have been able to travel, and a lot of change has occurred in demographics. So, and of course, jumping to 1900s, we know that The pandemic, the pandemic before the COVID pandemic was caused by Spanish flu or influenza, which was lasted for almost 20 years. And it was most devastating through World War I. And it's called the Spanish flu because Spain was not on either side. It was neutral, and they're the ones who called out this infection. And infections continued. They were transmitted because troops were being brought back from fields in camps and infecting more people and so on. So now what we learned from that time also was that there was an awareness of the transmission pattern of the flu. So that it was through the respiratory route, that there was a contagion that could be transmitted through contact, through respiratory contact, through droplets of people who had that respiratory infection. So to say, can you imagine that at that time, the Spanish flu wiped out 3 to 5% of the world population? So huge, right? Now, with that, we have developments in science where the organisms are being identified first through microscopes, because microscopes are first developed. And then I know Dr. Imran has a big interest in looking at wastewater surveillance. So see how important is that? Because identification of drinking water as a way of spreading diseases has been a continuous theme through spread of infections through the cities. And in fact, it is only when new systems for clean water and management of sewage was put in place in cities, in European cities, that the spread of diseases was actually reduced and it was contained. So now we leap to we're 2025 and we know so much more, but the principles of why we do what we do needs to be kept in our mind, because we have more advanced methods of trying to understand the way that diseases are being transmitted, the organisms that transmit them, and one needs to be mindful as to where something came from. So in your work, do try when you can take notes on where a sample came from. Something as important of that can be really, really important. I think that those of us who work in the lab, we treat everything as part of a, we take specimens as part of the course of the day, and often bits and pieces of data are missed. But if you want to make sense of anything, you need the information. So don't rush at that point, because it's so important. When was a sample collected? What was the source? And simple demographics of the individuals. Have they received any treatment? Because that will help you make sense of what you have. Now, what are we talking about here? SARS-CoV-2, as I said, I will use as an example. It is a virus. It is one kind of virus. There are many other kinds of viruses. We have DNA viruses, RNA viruses, and retroviruses. In principle, they all have a life cycle. They need to enter a cell, they need to multiply, and then they go through a lytic phase and they come out of the cell and they infect more cells. So they're organisms that are dependent, fortunately, on host cells to be propagated. They're also very easy to kill. If so, methods to clear viruses are actually quite straightforward, unlike bacteria, which have outer coats that are, resistant to many things, and they may persist. We can find bacterial DNA preserved inside bones, TB DNA, for example. So those, at the end of the day, it's your nucleotides. This is a double helical structure. It's the same for bacteria and for eukaryotes, but the principle is the same. One of them is, bacterial DNA is not is a different kind of chromosome. But the principle is the same, because there's base pairing that is exact between purines and pyrimidines, and it becomes important when we think of interpreting any information. We're looking at change. You've probably heard about sequences, sequence analysis, sequence interpretation. If you haven't done it, you'll be doing it. And you need to be mindful that just having variation in your sequence is not enough to determine if any change has an effect. So I put that out here because it'll be a recurrent theme. Initially when we had the ability to sequence DNA, it was very exciting to identify any base pair change. We believed it was a mutation, but now we know otherwise. We know that there are variations, and there are natural variations in the host and in all kinds of organisms that are related to lineage, that we work back to phylogeny, that we work back to things that are there but don't actually have an impact. And anything that is impactful or can be called a missense variation or a non-synonymous change is something that has an effect because it has a change in the amino acid that is transcribed. Okay? So a mutation is something that is caused because of a missense change or a non-synonymous change. And even then, it's not a mutation until it has a functional effect, in which case we call it a pathogen mutation. So when you have sequence data, how do you interpret that data? You will have so much information. I imagine they'll be going through bioinformatics, yes? And when you have the bioinformatics, you will learn that there's so much variation, you get so much data, but when you sort it down to what is functional, it becomes more manageable. And then, of course, at the level of the gene, where's that change occurring? It's important to keep all of that in mind. So, for example, this is just a schematic here of the same thing, that unless you have a base pair change that results in a change of the amino acid, there is no impact on the protein, okay? So here you have A to C changed, and this amino acid changes from A histidine to a proline. Okay, so epidemiology is defined as the study and distribution of health-related outcomes, and how do you use them? And then genomic epidemiology, which is what you are learning here, is the use of pathogen genomic data. And you can use it to understand distribution of organisms, spread in the context of infectious disease, and in the population. So where is it spreading? And how do you apply it? Okay, you can apply it to see just simply what the transmission pattern is. It can lead to studies that allow you to relate that genotype to a phenotype, because if in that case you're trying to see if there's a phenotypic variation because of that genotypic change, the phenotype may be some advantage of interest, which could be its ability to grow in a particular environment, to develop drug resistance. So anything in particular. So I'm sure that all of you are familiar with the PCR, which is the first thing one uses in the lab. It's the most clever, versatile, and simple method. And the real-time PCR is great because, used well, it can give you so much information. And as you can imagine, the information that you have depends on a very clever design of primers. But even that, you cannot get to that unless you have information about the gene of interest, okay? So we have a lot of tools in our toolbox, but to actually use it, you need now a lot of information about many genes and many targets to actually plan something that's more effective. So if you're looking at, you need to know the organism of interest, you need to know your gene of interest, you need to know within the gene, the region of interest. All that information is now available to us in public databases, fortunately, because over the last 10 years particularly, there has been a huge amount of data sharing, public data sharing, where if you want to publish something and you've used sequence information until it's publicly available, they will not publish it. So it's now conditional to that. So there's a huge amount of information. So even those of you who are unable to do vast amounts of sequencing yourself because of access, because of lack of resources, you can have a lot of learning if you know what you're looking for. And so just putting it out there. And then the idea being that if you know what you're looking for, then you can scale down and ask your questions even more specific. So just a show of hands, how many people have been looking for bacterial genomic markers in the past? Okay, just one. And what markers were you looking at? 16S. Okay, great. In what bacteria? Okay. Has everyone heard of these typing methods? You understand what typing is? Yes, loosely. So any variation, if we're looking at genomic epidemiology, we really want to know how an organism is changing. Okay, once you've speciated it, you've identified it's a particular species, that's step one. But then you want to look at what variations there are, if they're all the same, and you may want to do it for this exact reason, for a reason of transmission. Perhaps you want to know the relatedness between people, an outbreak investigation, or just try and understand the evolution, because you may have something that's spread from one region. Is it from a particular region? Because we know that natural variation occurs regardless of the phenotype, because the natural variation may be because there are families of bacteria or viruses, for example. So anyway, you can look them up later if you like, but there are points of variations which are multiple locus sites that are variable. We have them in all organisms. And MLST, 16S RNA, these are just variations in particular region that can be used. And these are things that have been done for many, many years using simple tools. And then you can use them using sequencing-based methods, Sanger sequencing-based methods if you're looking at particular regions, and then, of course, next-generation sequencing. And then, of course, I have to put in TB, because for TB, I've spent a lot of time working in tuberculosis, which is another old-world pathogen, which doesn't seem to go away because it's so clever. It can evade the immune system. It can become dormant. We have a whole problem of high rates of infection and persistent TB. And it has a genome that is very complex. It doesn't have any plasmids. It has a chromosomal genome, and it has more than 4 megabases in the genome. So it's a very large, complex genome. So it's a classic example of an organism that has a really tough outer waxy coat that prevents desiccation. And we know that these alterations in the genome really lead to drug resistance. So it's extremely clever. You need many drugs to treat it. It grows really slowly, so you need a long time to treat it. And then it keeps changing its genome. So I put this up here because maybe you learn to run a phylogenetic tree, but the idea is that if you have organisms from different regions, there are ways of looking at what the sequence similarity is, and this is how they determine what the fingerprint, etc., is of the organism. So I've already mentioned this. You can identify mutations. Now, we can jump to SARS-CoV-2, our current pandemic causative agent, believed to be the novel coronaviruses 2019, not believed, named as, and believed to be so novel that everybody thought the world would end, and hence there were lockdowns, and it was entirely not recognized initially to be related to either SARS-1, which caused an outbreak 10 years ago, or any other coronavirus. But we have learned more from that. Focus on one part of it, which is it's called a coronavirus because it has a glycoprotein coat that looks like a crown. And the spike protein, anybody who's heard of SARS-CoV-2 and is related to laboratory science has probably heard of spike. Spike is an outer coat protein. Very important because it is the protein that is attaching to host cells and helps the virus enter the cells. And also there's variations in spike. And because this interaction is so important, a lot of the variations that changed the disease transmissibility and the capacity of SARS-CoV strains as they evolved for the pandemic was because of variations right here, okay, in this region, the spike region. So a lot of developments in science occurred through the pandemic because there was such a need to understand how COVID was spreading. So our group and Dr. Imran's group and many others across the country were involved, even in Pakistan. I know NIH was very involved in sequencing efforts. Understanding transmission and understanding the variants as they came up was a very important piece. To know how things were spread, many complex models were set up to try and understand how transmission works. Fortunately, most of them were wrong because I believe for Pakistan, it was estimated that there'd be 500,000 deaths. That was the original estimate, but fortunately, it was wrong. And we'll see, does anyone know what the number is? How many people died of COVID in Pakistan? Reported deaths, kitne hain? To date? Nahi, Pakistan ka bataiye. Okay, so think about it as 31,000. Okay, 31,000 only compared to 7 million global deaths. So big difference. You've seen these platforms. Whole genome sequencing needs to be done for an RNA virus. It's a small virus, fortunately. So once the protocols were optimized, optimization was a bit complex because initially we didn't have the tools to do it, but once it was done and it can be analyzed, the sequencing can be done on short read and long read. Are they seeing short read or long read in this workshop? Short read, okay. So you know everything about Illumina, and Dr. Furqan told you about short read. The long read are smaller portable sequencers. So the Pakistan situation, so I will tell you a little bit about work from our lab and work from AKU in the context of the pandemic waves. And just to tell you that here we are, 31,000 deaths, it's up 31 now, and 1.5 million cases now. The number of cases that are reported depends on how much testing was done. And we know the testing capacity is limited, right? But the number of deaths, one can still be off a little bit, but it's not going to be 1 million or 500,000, right? It's 31,000. So what's going on? So that's where understanding the immune system in the context of variation in the virus was important. So here we are. This is our lab in molecular pathology to doing PCR-based diagnostics through the waves. We did rather a lot, especially the first 2 years, 500,000 tests. And so this is data from our lab that mirrors what was going on in Pakistan. And because of that, the efforts that we had in collaboration with the Department of Health and also NIH because we got samples from airports, we got samples from the initial cases that were identified across the province, because AKUH was key to identifying the cases and part of genomic surveillance. We learned some really interesting things. So the first cases we have were from different countries, and it shows that, before there was a restriction on travel initially, the initial spread was from cases all over the world, wherever they were bringing their samples from. We had the Zaire, Iran, Iraq, we had the Middle East, USA, Canada, many countries. And then what evolved was nomenclature. Okay, the virus was changing, but how do you try and identify changes and associate them with anything? What alphabet do you use? Because there wasn't nomenclature before. So the nomenclature for SARS-CoV-2 has also evolved, because there was pangolin lineages, and then they ran out of the lineage names, so they went to next clade lineages, which were more numeric. So that's why we have, pangolin lineages, meaning were just A and B, and then there was next clade, which was 1920 with ABC, and then there were other clades that we know were related to the Greek alphabet, which were Alpha, Delta, Omicron, but then they ran out. So then the virus keeps evolving. The point is that you need the tools, you need the sequencing, you need the bioinformatics to assess relatedness, because perhaps it was most likely the most well-studied virus, but certainly it was a very rapidly evolving virus. Okay, so what did we learn? One of the things we were able to do, and I put these up as an example of how in your own work, you may want to look at your data sets differently. So in addition to phylogenetic analysis, which is used to determine relatedness, one can look at rate of change, which is entropy. Okay, entropy means how many mutations are there across a particular region in a particular time frame. So we looked at entropy in genomes across the early pandemic period, 2020 and 2021. And not surprisingly, we saw that there was a lot more change, more variation, because you have a virus that is highly transmissible, but it's also trying to adapt to the population. And we know that the way the organisms work, they develop, there's poor, how do viruses have so many options of change? It's because they have poor proofreading. When they replicate fast, but then they don't have the error mechanisms that eukaryotic organisms do. So it's the virus that makes the most and survives the most, that then persists, right? So we have conditions where some viruses are selected better than others, okay? So I've said this before, that we have a database for information. We learn about alignments. AKU collaborated with the Government of Sindh, Department of Health, and we were able to learn about these. So one thing we did, and this is again something that can be done, I don't know what kind of bioinformatics we'll do, but you'll do in your course, but this is Bayesian analysis, where it's not just phylogeny, but it's looking at relatedness, and there's something called Beast analysis that we did in collaboration with a group in NIH USA that was providing genomic epidemiology training. So that was very useful. And this allowed us to identify, for example, where was the last most matching sequence of a particular individual. So it's not just countries, because there are ways that you can look at relatedness within strains, but you can also ask the question, what was the introduction? Okay, introduction means of that particular organism, where and when was that first type or virus introduced. So in this case, this was work where we identified the B117 in Karachi at the end of December 2020, and we know that the Alpha variant was, emerged at the beginning of 2021. And there was one case that came from Bahrain, and this was a person traveling from the UK, and they just had, a layover, just a layover at the airport. So we have stories, and this is just one example, but if you read, and I'm sure you have all those fantastic studies where they've done studies in transmission, like the Japanese ship that was left outside the Princess, was it the Tokyo Princess? That was where people were left on a passenger boat off the coast of the Pacific for many weeks, and because they were too scared to bring them on board on land, and they were able to show that transmission occurred through vents, and they occurred, and then other transmission outbreaks that occurred because people fed themselves to the same buffet because everybody's handling spoons. And then there were some amazing experiments where people were given money to go and mingle in closed spaces, like a party or a disco. Young students participated. Do you remember those studies? Amazing. They're looking at transmission dynamics. You give students lots of money, and they'll go anywhere. Okay, so we looked. That was the Alpha variant. We looked at it for Omicron, identifying BA.1. This was Ali Raza's work, where first there was the introduction to Omicron in Pakistan, BA.1, and then very interesting, through this kind of analysis, he and Javeria Ashraf, who did the bioinformatics piece, were able to show the way the introductions for each of these variants occurred in the provinces. So you can zone in through these complex bioinformatics and identify that, was the virus in Balochistan, from Balochistan, or did it actually travel from Sindh or somewhere else? And at least in the cases, now this was of course very limited to the samples that we tested in our lab from AKU. So this is not national database information. So it's a limited sample set, but these are examples of how you can use your genomic epidemiology data to ask questions of interest. Okay. And then also, again, think about entropy, which fits, looking at rate of change, where in 2020, there was less change across the genome. And then 2021, where you have all these emergences, alpha, beta, gamma, delta, and then ending with Omicron, there's change across the entire genome. And then it settles. Okay. Now, how do we relate it to vaccinations, and in terms of how the host responds to infections? That's the other piece we were looking at. One of the first things that we did was a small study based on serological sampling of healthcare workers at AKU, where we did get the first vaccinations in February 2021, and then we followed a cohort over 6 months. And Dr. Imran and the team had looked at serosurveys. You'd already seen how transmission's occurring in the population, so that's going on. You know that serology is an important tool. You can use antibody responses. So these are key antibody responses to spike, because we know that spike response is important, because that is important for the virus to enter. So if you have antibodies against spike, the presumption is, and then we proved later, as did other people, that you can stop the virus entering. So from here, we learned a couple of things. One of them is that there was very little information on inactivated vaccines. So BBIPB core V was what we received, and that there was a rate of antibody responsiveness in those that were over age 50. Now, effectiveness of vaccines that, test negative studies were done by AKU, one by Dr. Imran and one by Dr. Onab here, essentially test negative studies looking to see what the effectiveness was between different vaccine types. So, because remember that there was a lot of concern that the inactivated vaccine wasn't good enough and that we all needed mRNA vaccinations, but they were not available. And then there was the one-dose vector vaccine. That was another option. So how did they behave? So this is, you do this and it's great. We don't know what's happening in the level of the host. So another thing we did, and this was a big field study facilitated by the PEDS group in Matiari and Dr. Bhutta's group that facilitated the study for us, where we look at healthy subjects in a large cohort who received different vaccines, and we take samples at 6, 12, and 24 weeks. I'm going to present some data from that. And give you the summary. So it's on the top, that the waves are continuing. So the people we're sampling are healthy individuals. Okay, so everybody's healthy. We're not talking about people who have COVID here. We are looking at host immune responses. So the question we're asking is, have people who've received vaccinations of different types, and there are 4 groups here. A is inactivated vaccine, B is mRNA, C is one-dose vector, which is your CanSino Bio, and D is a group where there's the mixed bag of 2 vector dose vaccines, such as Chadox and Sputnik, because if you got those. And so we have, I'll take you through this so it's not too much information, but if we look at the top, essentially you see a rise and a fall. So this is A, B, C, D. You see this here, this is an outlier. So this is mRNA, the pattern is different. So mRNA vaccines give you a huge boost, and then that boost comes down, right? While the other vaccines also work, we knew they worked. But look at their IgG response to RBD, it's a bit slower, right? So hence, in parallel, we have seen this anecdotally, that there are more infections. Doesn't mean people have more disease, severe disease, but infections occur. Doesn't stop, vaccines do not stop people getting infections. And there's a period in which there are most infections. We have some other work that showed that after the inactivated vaccine, it was the 8-week period, which again is this period. Okay, the early period where they're most likely to get infections. Okay. We also did neutralizing antibodies. So neutralizing antibodies. Now here you can look at the viruses. So we did pseudotype particle assays where we had pseudotype particle to wild type, Delta, and Omicron, and compared vaccine efficacy. And the idea is that you infect the cells and you look to see if the GFP particles that are carrying the virus or the pseudotype virus, which is essentially the spike, and the spike protein can be from any genome, but it has a GFP tag. So if it goes into the cells, then it glows and it shows infection. Many of you who've worked in virology labs must be familiar with plaque assays, plaque production assays. In principle, anyway, they're hard to do. SARS-CoV-2 is a BSL-3 pathogen if you grow it. So it was challenging, but very useful to set up a pseudotype viral particle assay because it gives you a handle on the sample you have, which is now a serum sample of people vaccinated with different cohorts. And, the curves, neutralizing curves have to be interpreted, and then we get data that is related to % inhibition of entry. And we see that, in fact, Sinovac works, again, wild type. By 6 weeks, you have a robust neutralizing titer and then goes down. mRNA-2, CanSino Bio is lower. And then in the comparative group, we saw higher neutralizing titers after mRNA vaccine. What do you think happened when we tested the same individuals vaccinated in 2021 against Delta pseudovirus? Any idea? What will be the next result? Anybody? Tazeen. You lived through the pandemic. Okay. Now don't be shy. So we have higher neutralizing titers against Delta after mRNA vaccines. So other people have also seen that. So these were people, there could be two reasons for it. One, that even though the vaccine was the wild type COVID vaccine from 2021, it was important to do this work because Pakistan did not get updated vaccines. What you had was what was given, which was the original wild type. And in a lot of countries, the newer vaccines were updated, but we didn't have that privilege. At the most, booster vaccinations were given. So this protection may be because of the mRNA. That has also been shown that the mRNA somehow was more effective against Delta, but also because there was transmission in the population. Okay, so the transmission, if you have infection and you control the infection, then you will make protection against Delta. And then what we did was we did a pre-test. So now we said, okay, we've got the same group. We've got 3 cohorts here. We've got inactivated mRNA and vector vaccine cohorts. And here we tested them against the JN1 Omicron, which only emerged in 2024. So completely unrelated. And in fact, we don't see the titers. Okay? So that tells us that why the COVID infections continue. They persist because you have viruses that are emerging where at least their spike region is different enough that the earlier humoral response does not respond. But how is it that we are protected? It's because the T-cell responses are more consistent. Okay, so we looked at T-cell immunity after 2 kinds of vaccinations here. This is inactivated and mRNA, and they were comparable. Okay, so they're still there. Any questions here before I go to the last piece? ये cellular response है, ये humoral response है. जी, sorry, I moved too fast. हमने cellular response देखी. Or cellular response देखी, specific to a new kind of assay, which is like QIAGEN had made a COVID IGRA assay. And the COVID IGRA, maybe you've heard of it, was a 5-tube assay where they had, have you done IGRA assays? The quantiferon IGRA assay? Okay. So it's a whole blood assay where you expose your samples to different antigens, and then you measure the T-cell interferon response. Okay, so if you have a sample that has T-cells that are recognizing the antigen in the tube, you make interferon. So what we had is an assay that was measured a spike response as well as a response that was the whole genome, so E and M protein. Because although your humoral response tends to be specific to the spike region, your T-cell response can be broader because there's so much more in the SARS-CoV-2 virus, other than spike. And which is why in those other regions vary less. So the variability across the genome and how they're changing also links back to what your memory T-cell is remembering. Okay? Okay, anything else? Anybody else? Okay, I think that's quite a lot already. So my last piece is give you an example of how you can. So sequencing is great. I think you're already converted, which is why you're here, and you'll learn a lot, and you'll see how you can utilize the genomic sequencing information and apply it in your own fields. As I said, you don't have to do all the sequencing yourself because there's so much information available in databases. And, you've got star bioinformatician, Dr. Vakas here, who will be guiding you through different methods. And here's an example of how many things and how many applications one can have. So, for example, I mentioned that we had done initial sequencing for SARS-CoV-2 in 2020, but there was a shortage of reagents. There was trouble with the machine. There was a lot of wait time, but we didn't want to completely stop trying to identify these strains because we were getting the samples through our health surveillance, and we were committed to doing whatever we could to inform the public as to whether it was, at that time it was a big deal. Is it alpha? Is it delta? Because there was no vaccine available at that time. So how did we do it? We went back to using kits were available, but we could have done it using local primers for signature mutations. So this is my comment on the signatures. If you know what you're looking for, and the spike region nomenclature was changed to identify different variants of concern. For example, it was very easy in the beginning because alpha was identified by N501Y. Then there was more mutations associated later with vaccine escape, E484K and K417N. So quite simple 2-3 target mutation-based approaches were used by us, by other people, to identify the emergence and the change in virus types. Okay, things like, so in fact, in the UK, a lot of the work done by Genomics England, the Public Health England, was based on using a coincidental assay, which was the thermotac assay for SARS-CoV-2, because there was a region that was different in alpha compared with wild-type strains. So it was, if you know what you're looking for and you're familiar with what the change is, you can come up with intelligent approaches to track changes. And so, for example, we were able to track changing variants in Karachi in Pakistan from alpha to delta through the waves using assays like that. So we saw changes. This is blue, which is your older strains, and delta is yellow, and it comes up, and then you have change of Omicron at the end. So, and this is using, so we continued to do the genomic-based whole sequencing confirmation for some samples, but we did hundreds of tests using simple assays. Okay. This is the last example I'm going to give. Many of you work in clinical microbiology, and we know that drug resistance is a big problem. Perhaps some of you have come here because you want to be able to apply your genomics training to understand and improve the way that you diagnose resistance. So M. tuberculosis is a good case for identification of resistance confirming genes because it needs many drugs to treat it. And for rifampicin resistance, it's the RPOB gene, which is a region which is very discrete. It's only a 100, 200 base pair region. That is the one that has most of the mutations. It's in a cluster. So hence you have diagnostic assays that were traditionally identified to have a simple PCR with a TB sequence plus an RPOB 531 mutation. And there's a lot of information regarding the complexity of whole genome sequencing, but also the interpretation. So I'm not going to go into that, but at least in the TB field, there are many bioinformatics tools that are available to process data, to identify the genotype linked with the phenotype that's been observed, because the genotypic variations are very large. There are many drugs that are used in your primary treatment, you start off with 4 or 5 drugs, and then you change them when it's MDR, and then more if it's XDR. And we're learning more and more about how the different drugs in the TB pipeline are working or not working. And there's variations related to where the bacteria are from. So lineage variations, that's a whole different story. Then you have drug resistance associated variations. So, if that's a space of your interest, then there are many things that you can learn from here. So essentially, once you have the information, you will learn how important bioinformatics tools are, how you deal with FASTQ files, what BAM files are, and then you'll be able to go through different protocols that allow classification and then any other thing that you may want to do. So this is just showing the same. There, as I said, there are tools that actually have ways of sorting phenotypic versus drugs versus targets that let you select and let you identify resistance. So this is my last slide, and I end here. And the work I presented is, thanks to a lot of collaborations and people from the department, my group, other groups, collaborators. These are just people who are involved in the COVID work. And thank you for listening.
I will be making talk, and I think you saw in real time the power of pathogen genomics, and I think everybody will agree that SARS-CoV-2 was probably the best stage virus so far, and it's kind of then also brought the genomic epidemiology to the forefront as well. So, as you were talking yesterday, it's any understanding of the infectious diseases is now incomplete without incorporating the genomics component into it. So, any questions for Dr. Zahra?
I showed you my lessons. These were the lessons we learned.
COVID has actually challenged our capabilities, and it has actually made us realize that how multiple approaches can be implemented and how things can be mobilized, and the world that could have that we thought would have happened in 6 months would have happened in just fraction of time. This is what I have learned. This is my perception, my understanding that we have learned. So actually, we were also working on COVID, and we were actually, I was on a team who was actually involved in vaccine. I was at that time I was serving the distro, and we were the team who actually developed some candidate vaccines, vaccine candidates for COVID-19, and in parallel to that, we were working on the genomics and everything. So, and I understand because Alpha is very well developed facility, but our facility was actually developing the one thing, and I guess that COVID is a blessing in disguise for us, and also for so many other institutions. And it has actually taught us a lesson that we have to become self-reliant in so many things. And now it's the time to, if we are standard, now it's the time to upgrade ourselves and to implement and to compete with the developed world. So it has gone so long. I just want to
Okay.
give your perspective.
Thank you for sharing that. absolutely. We need to develop ourselves, our laboratory sciences. But I think key is the realization, and I hope that it stays, of the appreciation of laboratory science. If there would be one thing, the realization of how important it is to invest in science. Because we had a situation where the people treating patients were unable to fully understand the disease biology. People administering vaccinations were also a little bit in the dark, but there wasn't enough. I still believe there just wasn't enough attention or belief or appreciation of the scientific work and how it would inform medical practice. I think we remained, we had a relationship which was a functional relationship at most, but it was not truly appreciative, I think. And I think that there has to be a realization that you need science, you need genomic epidemiology, you need immunology, you need vaccine research. It's not a joke, and it's not a luxury. I believe that the realization that it's not a luxury is a key thing that is still missing, because we can bring in vaccines that have been tested elsewhere, but how effectively are they working? Just by giving them and then seeing how people get sick is not a good way, is not an ideal way, shall we say, of seeing how effective vaccines are working. You need the immunological analysis to understand the impact, because otherwise you're essentially playing with people's lives, because you give them a vaccine and then you say, oh, how sick did they get? How many admissions did you have in the hospital? Is that a good way? Ideally not. So I think that that will be one hope for me. I learned a lot in terms of what we need to do further. Over to you.
You can relate it that all the historical context that Dr. Zahra also presented that, people were doing a lot of different things over time, but understanding, as we started with something very recently that what actually we are doing about the biological process of course. So I think it's very important to understand from that perspective. Any other comments, questions?
Hasan, what do you think? Since the spike protein is highly evolving, yet infections are not as deadly as they used to be, because T cells have a memory of the virus that is not specific to the spike protein. Do you think that the inactive vaccine, which was administered before the mRNA vaccine, was more effective than the advanced one in the long term?
Okay, so the virus has become endemic, which means that it continues to circulate, but people are less sick. Now, I didn't have time to go into the vaccine biology of everything that we've looked at. But we, like other people, have observed that your immunity is lower in high-risk groups and in people over 50. That is the natural biology of T-cell aging, okay? So we had a spike earlier this year. In fact, we had people in hospital, and then we identified the JN1 with your group. And that identified that, the virus is there, but then we looked to see who was it who was in the hospital, in the ICU with that virus. And it was the immune compromised. So immune compromised individuals are the ones who will get influenza. They will get acute respiratory infections. So you can't stop that. Okay, so that's one thing that you will not stop to have. You will continue to have circulation of this. Now, comparison of long-term effect of inactivated and mRNA vaccine, that's something we are looking at right now. We are looking at long-term. We don't have that data, but I think that it's obvious that it does work, because what we have seen is that the humoral response continues. We have looked at people a year and a half and now 3 years after vaccination, and they still have a response. What we don't know if that response is because of continuing transmission in the population or because they have a memory response, likely both. So in short, inactivated vaccines work. Yes. Thank you.
So we are going to go in the same way we went to WCS yesterday. Okay, so the posters are there waiting for you. You can freshen up and just gather downstairs. Okay, and we'll meet upstairs, first floor, WCS for lunch. Okay. Okay. Thank you so much. Will you go to questions from the bioinformatics part for setting up their laptop? Anyone else? Sorry, only one question.
Okay, so no, it's good. But say something.